Hi, and welcome to the final session of this today's UFAW conference. My name's Claire Witham, and I'll be chairing this final session of the day. To start, we have a presentation from Grace Fuller of Detroit Zoological Society, and please do feel free to ask questions throughout the talk using the questions box.
Hi, my name's Grace Fuller, and I am the Director of Animal Welfare for the Detroit Zoological Society. And I'm excited to speak with you today about a new line of research that we've been embarking on, exploring oxidative stress as a potential indicator of animal welfare. So I'll start with a brief review of oxidative physiology for those that might be less familiar with it. This is a relatively simple figure, but what I like about it is that it conveys that the crux of oxidative physiology is a balance between positive and negative factors, much like animal welfare itself. So reactive oxygen species and oxidative compounds form as a result mainly of normal cellular metabolism. This is the price we pay for being aerobic organisms that breathe air. That process generates reactive oxygen species and free radicals that have the capability to damage internal cellular components. While some of these are derived from the environment, from UV radiation, for example, the bulk of them are the result of our normal cellular metabolism. We also have in place antioxidant defenses, which serve to neutralize those oxidative compounds and prevent the damage that they can cause. Some of these are endogenously produced, while others are sourced through our diet. But the upshot is that when coupled with the activity of enzymes and other repair mechanisms that reverse the effects of oxidative damage, our antioxidant defenses help us to more or less neutralize this damage and maintain homeostasis with little damage occurring. One of the many factors that can influence oxidative physiology is the activity of glucocorticoids, which of course are of interest due to their central role in the physiological stress response and their ubiquity as a measure of animal welfare as a result. However, glucocorticoids play many regulatory roles within the body. They're always performing their metabolic roles, and under normal circumstances, their effects on oxidative physiology are relatively minor. So when you increase the expression of glucocorticoids, there is an increase in the metabolic rate, which means there is an increase in the production of reactive oxygen species with their damaging potential. However, under these circumstances, there is a compensatory measure which kicks in, and antioxidant activity is upregulated as well. And as a result, the net effect is more or less zero. Homeostasis is maintained, and there is no major source of oxidative damage occurring. Although the effects of glucocorticoids are relatively minimal in the short term, under conditions of chronic stress or when there is heavy activation of the HPA axis, glucocorticoids can contribute to the state known as oxidative stress. They're one of many factors that can cause oxidative stress. And so in this case, what is happening is that the metabolic rate and glucocorticoid activity is increasing so much that the antioxidant system is no longer able to keep up. And the result is that oxidative damage starts to occur, wherein you have damage to important cellular macromolecules like lipids, proteins, and DNA. Studies of oxidative stress in human medicine have been going on for some time, but it's only more recently that people have started to investigate oxidative stress in non-human animals outside of laboratory models to understand things like behavioral ecology, to understand how oxidative stress interacts with different strategies to survive and reproduce in the wild, or to look at how oxidative stress is impacted by housing or other management practices related to caring for animals in captivity. And so I'll talk about some of these different studies using Meller's 5 domains model to sort of organize the different results. And also, of course, noting that I'm sure there are many omissions because I couldn't possibly cover all of this research in this span of this talk. So combining the nutrition and health domains here, there are studies linking oxidative stress parameters to feeding strategy in wild Gen 2 penguins, also linking oxidative stress to the diets of farmed carp or consumption levels in body mass in captive parrots. There are also studies using oxidative stress as a parameter, looking at dogs, domestic dogs in shelters, and its relationship with different illnesses occurring there, as well as looking at oxidative stress in relation to lameness in dairy cows. Moving on to the physical environment domain, there are studies linking oxidative stress parameters to ecotourism intensity for marine iguanas. There are studies comparing oxidative stress parameters based on overall environmental quality, both in comparing cheetahs in the wild versus in captive settings, and comparing different qualities of the environment for wild boars. There are also studies linking restraint stress to oxidative stress in cheetahs, to anthropogenic effects of noise on tree swallow nestlings or group size in alpine marmots. There are also studies linking oxidative stress to both cold and heat stress in non-human animals as well. So many aspects of the physical environment have the capacity to affect this system. Moving on to the behavioral interactions domain, oxidative stress has been related to crib biting in horses, which is often considered a negative indicator of welfare or stereotypic behavior. Oxidative stress is also related to social interactions among animals, and that includes the dominance rank of mandrills or the ascent to dominance in cichlid fish. And oxidative stress has also been related to variation in aggressive behavior in wild birds. Finally, looking at the affective domain, there are studies linking oxidative stress to coping style in alpine marmots, as well as personality in laboratory mice. There are studies that link oxidative stress to a variety of affective disorders in humans and that show elevated levels of oxidative stress chronically in humans that are exposed to chronic stress. And of course, I'm not the first person to suggest that there could be utility for these measures for understanding animal welfare. So Brambilla et al., for example, looked at oxidative stress as a measure of animal welfare in farmed swine. One of the complexities of studying oxidative stress is the wide variety of biomarkers or targets that one could be examining. So you can measure oxidative compounds themselves. This involves measuring free radicals or reactive oxygen species, reactive nitrogen species, or perhaps their metabolites. But one of the difficulties of measuring these types of compounds is that they are inherently unstable and therefore difficult to measure. They don't store well, so accurately measuring these kinds of biomarkers can be a little bit challenging. Other studies have taken the approach of directly measuring levels of antioxidant compounds, or you can measure the overall antioxidant capacity to absorb oxidation of blood, for example, or you can measure the activity of the enzymes which catalyze the redox reactions which serve to neutralize reactive oxygen species. Other studies have taken the approach of looking a little bit further downstream and measuring oxidative damage directly, so getting past the point where it's been neutralized and instead measuring the byproducts of damage to lipids or to proteins in the form of carbonyl groups usually, or to DNA itself. Studies of DNA damage in humans go back a while, looking at the relationship between DNA damage and the development of cancer. This is a pretty well-established relationship that you may be familiar with. However, studies in non-human animals, again, are a little bit more novel. One recent study showed a relationship between handling stress and rates of DNA damage in house sparrows, which suggests that DNA damage does have potential as a measure of acute stress, that it changes acutely in relation to those types of events, in addition to serving as a measure of chronic stress. Studies in humans, again, have linked DNA damage specifically to disease states, including major depression and schizophrenia. And epidemiological studies are increasingly employing DNA damage as a measure, in part because it can be measured non-invasively in urine. And these studies have shown relationships between things like self-reported levels of stress at work, other lifestyle factors such as smoking or activity levels, and rates of DNA damage. So we are very interested in measuring DNA damage specifically because of its relationship to telomeres. So telomeres, you may be familiar with, are the non-coding ends of chromosomes, and they are particularly vulnerable to oxidative damage in large part because of the guanine-rich nature of these sequences. Guanines are particularly susceptible to oxidative stress. And in fact, telomere shortening reflects chronic oxidative stress is occurring. Um, However, if you don't have the ability to measure telomeres directly, measuring DNA damage could give you also some indication of the response to acute changes before the long-term damage to telomeres occurs. It's also something that's relatively easier to measure in a lab. Using enzyme immunoassays, we're able to measure DNA damage in our lab, although we do not have the facilities to do genomic techniques. We can do this using endocrinology essentially. And it gives us the potential also to explore means of non-invasive measurement. As I mentioned, DNA damage can be measured in urine, and there's reason to believe that it can be measured in feces as well, although to our knowledge, there have not been many studies or any that really have employed this technique. And of course, so this, of course, brings us to really the work that inspired these studies, and that was Melissa Bateson and her colleagues' works on telomere shortening as a cumulative indicator of animal welfare. And so, again, as they pointed out, that telomere shortening is related to a variety of different organism level experiences, right? So it's something that's influenced by stress, by positive factors, by negative factors, all of which are summed together to result in this overall measure of biological aging, which essentially functions as a measure of life quality or as a record of how much chronic stress an animal has been exposed to, with the expectation that there is this accelerated rate of biological aging that accumulates as a cost of stress. And so looking at this figure, you can see here how the organismal level experience is filtered through the cellular environment. And so the area that we're targeting here is oxidative stress, which of course is influenced both by cortisol and by some of these positive and negative organismal level factors. And so one way to get around the sort of complexities of that, because it suggests, in some ways that it's a useful biomarker, the fact that it responds both to positive and negatives, but that also suggests that it could have complexities in its interpretation the same way that glucocorticoids do, where it's not always clear if the changes represent a positive or a negative impact. One way to get around this, again, is to measure the biological cost itself, and in this case, by measuring directly the damage done to telomeres. And so that was the inspiration really for our studies of DNA damage, with the expectation that overall, when we saw higher rates of DNA damage, that would be reflective of greater damage to telomeres themselves. So here at the Detroit Zoo, we've started doing some preliminary testing to evaluate whether fecal DNA damage could be a non-invasive biomarker of animal welfare and sort of a cumulative measure of animal welfare. And so our goal has been to assess evidence for using fecal 8-hydroxy-2'-deoxyguanosine, or 8-OHDG, which is one type of DNA lesion, as an indicator of welfare. And in order to do so, we're looking at correlations between the DNA damage measure and fecal glucocorticoid metabolites. And then we're looking at relationships between DNA damage and behavioral indicators of welfare as well. And so we have two different case studies where we have investigated these relationships using grizzly bears and white rhinoceroses. So our first case study was undertaken during a construction process where we were expanding the home of our grizzly bears from that green shaded area to the overall area outlined in green, which however did necessitate a stay in the smaller area outlined in purple during one portion of the construction process. So at that point we had 3 large adult male grizzly bears all living in a somewhat restricted space, and we thought that this compression of space and the construction itself would be sources potentially of stress, and we monitored the bears using fecal samples and by collecting behavioral data. So we validated the assay for cortisol in feces using an enzyme immunoassay sourced from Arbor Assays, and with in-house coded plates. We have good validation data for this assay, as you can see, and we analyzed 17 to 37 samples for each of our 3 bears. We also had good results validating the assay for DNA damage in the grizzly bears. We measured it in the same fecal samples. Again, we have good parallelism recoveries and good CVs indicating good assay performance. So I'm going to look here at longitudinal profiles of both FGMs and DNA damage for the bears and how well they correspond. You can see here the FGMs for our first bear, Boo, as well as the baseline and 2 standard deviations above and below the baseline. When I overlay the DNA damage measure, you can see that they are well correlated for this individual. So we're seeing a good correlation between DNA damage and FGM levels. Similarly, here is the graph for his brother Mike, showing his FGM levels, and then next I will overlay the DNA damage measure. And again, for Mike, we see a nice correlation between the two measures. Finally, we'll go through the same process for Thor. And as you can see, Thor had a really strong correlation between his FGM and DNA damage measures as well. And so when we look at this a little differently, the scatter plots again confirm that we have a strong relationship between measuring DNA damage and measuring fecal glucocorticoid metabolites in feces. And there's overall a very strong correlation for the bears. Another interesting thing that we found in this study was a relationship between rates of affiliative behavior and DNA damage. Now, this was more of a trend. It was not quite statistically significant, and the relationship was different for different bears. So for Boo, we saw that higher rates of affiliative behavior were associated with lower DNA damage, while Mike showed the opposite relationship, and Thor kind of showed no relationship. Although this is very preliminary, it's also very in line with what we would have expected knowing their personalities, so that was sort of interesting. So in conclusion, we saw a great correlation between FGMs and DNA damage in this fecal extracts in this study, suggesting that we have some good validation data for measuring DNA damage non-invasively. There did seem to be a relationship between DNA damage and social behavior, but this is a very small sample size and a very preliminary study, so we don't want to overstate that relationship, especially given that we realize many factors require further validation, including the diet, sample storage and processing, and the origin of the DNA lesions that we are measuring, where in the body exactly were they coming from. Our second case study was looking at visitor effects in our 2 southern white rhinos living here at the Detroit Zoo. We studied them during and after the zoo was closed to visitors for an extended period due to the COVID-19 pandemic, and using similar approach collected data on their behavior and measured FGMs and DNA damage in fecal samples. For the rhinos, we used a corticosterone assay rather than cortisol, and as you can see, we have good validation data for that. We had good validation data for the DNA damage assay in the rhinos, and everything indicating that it showed good performance as an assay. Again, kind of the same approach, overlaying FGM results with DNA damage results. You can see that for Jaziri, there's really no relationship here, and in fact, it's not statistically correlated at all, so those two measures were not related for him. Looking at Tamba, there is, in fact, a correlation between FGMs and DNA damage levels for Tamba, but it was actually the opposite direction than we were expecting. So he had higher FGMs when he had lower DNA damage rather than the positive correlation we expected to find. We also looked at the effects of zoo status, and while for Jaziri, we did see a significant increase in FGMs associated with the opening of the zoo compared to when it was closed, we did not see any relationship between DNA damage and zoo status for either of the rhinos. And I think that's largely due to the fact that there was such a high degree of variability in the DNA damage values that we measured, and this was even still after removing some really obvious outliers from the data set. We looked at behaviors in relation to visitors that we thought might be significant, and one of those was percent of time spent alert, which was positively correlated with DNA damage for Jaziri. However, we didn't see relationships with any other behaviors for him. So we think that FGMs and DNA damage, they were correlated weakly for one rhino in this study, but it showed the opposite relationship that we were expecting, and we think that the high variability in measuring DNA damage here is probably reflective of the challenges of measuring it in this particular fecal matrix and the large quantities of hay that were in it. And so we're not entirely clear if we reliably measured DNA damage in this case. Overall, we found a very robust correlation between FGMs and DNA damage in one study, but not the other. And we suspect that the quality of the fecal matrix made a big difference here. These data are very preliminary and a lot more validation is needed. And so, that's partly why we wanted to share these results, to encourage people to conduct these validation studies that will help us understand these measures. Because we do still believe that DNA damage has potential as a comprehensive measure of animal welfare. And we think that it has a lot of potential because you're measuring, in this case, a biological cost rather than measuring a chemical messenger like cortisol. So we think this has potential, and we're going to continue doing some of these investigations here at the Detroit Zoo. I would like to thank everyone involved in these studies, particularly our lab tech, Emily Bovee, and our volunteers who help with sample processing in the lab. And I will show some references briefly. Feel free to contact me for further information or for the reference list. And I just want to say thank you very much for listening. Bye-bye.
Hi, thank you. Grace, if you want to turn your camera and microphone on, I've got a few questions for you. Thank you. So first up from Georgia Mason, she says, very cool approach. Is DNA damage a measure of health senescence more than effective well-being though? Since telomere shortening, for example, is enhanced by sugar consumption, alcohol and UV exposure, and healthy, yes, but not unpleasant or stressful psychologically.
Yeah, it's a good question. I mean, one of the reasons that we think it has potential to be sort of a more comprehensive measure is based on some experimental research that I didn't go over in what was already a lengthy literature review. But for example, if you experimentally administer cortisol to a lab animal that's housed solitarily or in a social group, the effects of oxytocin kind of neutralize the effects of cortisol and prevent DNA damage in the socially housed animals, but not the solitarily housed animals. So there's some suggestions from other studies that this does have the potential to kind of sum together those positive and negative factors into a measure that takes both into account, but there's so much validation that needs to be done. Again, this is very preliminary work that we sort of wanted to share in the spirit of, hey, this is something cool to try out, knowing that a lot more validation is needed.
Thank you. I think we've got time for a few more questions. Heaven Browning asks, do you think the temporal correlation between FGM and DNA damage in the bears at least suggests it's an immediate rather than cumulative welfare indicator?
Right, and that's a good question too. And so I think that there's evidence showing sort of both effects with DNA damage, right? So in humans that have chronic conditions like depression or schizophrenia, you have this kind of chronic accumulation of DNA damage and higher rates of it that go on for longer periods of time. But then there's been other studies like with the house sparrows that show that a very acute effect, right? So I think kind of figuring out the time course of this is, again, more questions that need to be answered. You know, when we analyzed our data for the bears, we typically, we've done studies, to see kind of what the transit time was so that we can adjust the fecal glucocorticoid levels to compare to our behavior data. And we usually do like a 24-hour lag time to excretion for hormones. So we backdate things by 24 hours. We didn't know what it would look like for DNA damage. And so we made the assumption that it would also be 24 hours. And that makes a strong correlation between the two measures. But again, there really needs to be a study where someone, does like radio labeling of the DNA lesions to really figure out the time course that they're occurring in.
Great, thank you. I think it's time to move on to the next presentation. So thank you very much for your talk. You want to switch your camera off, thanks. So I'm going to hand over to Hugh Golich now for the presentation of the UFAW medals.
Thanks, Claire. Okay, I'll share my screen. So thanks, Claire, and thanks for the great talk just now. This is one of the more pleasurable bits of what's a very pleasurable meeting in general. It's my pleasure to give UFAW's awards for the year. So we give awards for both early career researchers and researchers who've made a lifetime contribution to animal welfare science every year. If you're interested in nominating someone for future years, please go and have a look at the website. But for this year, I'm delighted, first of all, to make the Early Career Researcher of the Year award. And this year we had a number of really excellent nominations, and in the end, the panel couldn't split the two best nominations. So we've decided to make a joint award to Dr. Jenyun Chow from the University of Pennsylvania and Dr. Jamie Aloy Dallaire from University Laval in Canada. If you'd like to pop up on screen, I can congratulate you in person. There we are. So congratulations to both of you. The panel was really impressed by what an excellent start you've made to your careers, not just in research, but also in outreach to get real world change for animals using your research findings, and in supervision of students and a number of other things. I won't go on to embarrass you both too much. If anyone else wants to read more about why these are such deserving winners, have a look at our website, ufaw.org.uk/ECR, where you can read the full citation. But I'd just like to say a really big congratulations to both of you. It's an excellent achievement. I'm sorry we can't have you there in person to accept your award, or indeed that we can't hear a round of applause for all of you, but congratulations, both of you. I'm delighted. So if anyone would like to hear more about both Jamie and Jen's research, they'll be giving talks tomorrow afternoon, so you can go along and hear more about their research. So once again, congratulations to both of you, and looking forward to hearing your talks tomorrow. It's the best we can do for applause. I'm sorry. Everyone else is clapping, you can't hear them. Thank you. So, it's now also my pleasure to move on to present the UFAW Medal for Outstanding Contribution to Animal Welfare Science, which is given for a lifetime of achievement in animal welfare science. And again, I'm delighted that this year the award goes to Professor Joy Mench from the Department of Animal Science at UC Davis. Joy's work embodies what I like to think is the aim of UFAW, which is not just to promote animal welfare science, but to make sure that that science gets used to benefit animals. So as you can see from Georgia Mason's nomination, Joy has done exactly that by both being a really important basic scientist who's improved our understanding of animal welfare, but also someone who's gone out to promote that in the real world with the industry, with policy makers and the makers of guidelines and so on, to ensure that animals really benefit from both her research and that of other people. And not only that, she's also an inspiration to the next generation. I plucked a couple of these tweets from Twitter from two of our previous early career award winners, or one who you've just met, showing what an influence Joy's work has had on them too. So that's another incredibly important reason why Joy is our winner for this year. So again, if you'd like to read a lot more about Joy's work, have a look at our website where there's the full citation for her award. But I won't embarrass her anymore except by asking her to appear on screen and accept her award, and then afterwards she's going to give a keynote presentation as well on the development of animal welfare standards, which should be really fascinating. So Joy, if you're there.
I'm here.
Fantastic. Well, congratulations again. You're an incredibly deserving winner of this year's medal, and again, I'm sorry that we can't be presenting it to you in person, but congratulations.
Thank you so much.
Can I say one person applause?
Can I say just a few words?
Absolutely, please do.
Well, first of all, I'd like to thank Georgia and Cassandra Tucker for nominating me for this award, and obviously UFAW for giving me the award. But I did want to say that when I went to the UK, the 40 plus years ago, to get my PhD in animal behavior, I knew I was interested in the science of animal behavior, and I knew I was interested in animal welfare, but I honestly at that point had no idea there was a relationship between the two things. And when I was preparing to do my experimental work in the UK, I went to a meeting on the 3 R's. And it was at that meeting that I became introduced to UFAW and the work that UFAW had done to improve laboratory animal welfare and was also doing to improve farm animal welfare. And that really is what sparked my interest in pursuing a career where I could apply my science to the improvement of animal welfare. So I owe a huge debt to UFAW. I have huge respect for the work that UFAW has done, and I'm just very honored to receive this award.
Thank you. Well, I'm honoured to hear that we had such an influence. So that's fantastic. So thank you again, Joy. Congratulations from all of us at UFAW and everyone at the meeting. And I think now we'll go ahead and hear your talk.
Okay, thank you.
Thanks, Joy.
Good afternoon again, everyone. This is me, pre-recorded this time with a few little glitches that I hope won't prove to be too distracting. It's a long title, but it reflects a lot of the things I've been thinking about related to the activities that I've been involved with during the latter part of my career, which is working with various groups to create meaningful animal welfare standards and auditing programs. My own work has primarily been with poultry, and you'll hear me refer throughout the talk to farmers or producers, but I really hope that the comments that I'm making and the points that I'm raising in this presentation will apply across a variety of sectors, regardless of whether you work with farm animals, zoo animals, laboratory animals, or companion animals. I asked Cassandra Tucker to co-write this presentation with me because she has been doing very similar work in the cattle sector, particularly for dairy cattle, and it's been very interesting for us to sort of compare and contrast the approaches that are taken in different sectors. But broadly speaking, we've identified 5 big pieces that are important to consider for animal welfare assurance programs. That there be clear goals, support on site for program implementation, that the audit has to have integrity, that the standards have to be meaningful, and that there has to be transparency and accountability. And these things have to fit together in a very clear way in order for an assurance program to be robust. And by robust, I mean that the program actually achieves its own programmatic goals. In this presentation, I'm going to talk about 2 of these pieces, meaningful standards and audit integrity. And these are very interrelated to one another. Each of these has multiple subparts. For example, meaningful standards have to be comprehensive, explicit, have realistic metrics, and be revised regularly. But I'm just going to talk about a small part of that in terms of how we frame standards, what is required, and from the audit perspective, how we verify those standards, that is, what is measured. We can identify 2 broad approaches to standards and auditing. The first involves specifying or measuring inputs, such as resources or management, and the second involves specifying or measuring outcomes, and those outcomes can be measured at the level of the animals, the humans, or the environment. And I'm going to talk about each of these in a little more detail. In terms of specifying resources, this is a very broad category. It could include things like housing configuration, feed, water, environmental enrichments, space, or social companions. Management requirements are largely record-keeping based. That is, they involve things like documentation of relationships with consultants or contractors, for example, veterinarians, nutritionists, emergency plans, veterinary health plans, standard operating procedures that are specific to the particular sector, personnel training records, etc. In terms of measuring outcomes, as I said, we can measure those at the level of the humans. That would be, for example, personnel competencies in performing their assigned tasks or human-animal interrelationships. We can also measure outcomes at the level of the environment, for example, air quality. And outcomes can also be measured at the level of the animal, and that can be either a very broad-based kind of assessment. For example, on the left, I'm showing the welfare quality assessment protocol for poultry. I'll talk about welfare quality a bit more in a minute. But this is a very broad-scale evaluation of health, physical condition, and various aspects of behavior measured at the level of the animal. Or we can be looking for an outcome that's very specific to a particular procedure or situation in which the animal finds itself, for example, in the assessment of pain. When we look at existing farm animal welfare standards and audits, it becomes apparent that they're very heavily weighted towards specifying and measuring inputs rather than specifying and measuring outcomes. We do see some hybridization, I think particularly on the side of measuring environmental outcomes. So it would be very common in a set of standards to see specifications for ventilation and how that ventilation is to be managed, but for the verification of that to be carried out at the level of measuring things in the environment, for example, ammonia or dust. I think there are historical reasons for this emphasis on inputs in animal welfare standards and auditing programs. I know we're all very familiar with the 5 freedoms. They've been extremely influential in shaping these kinds of programs. And I'm sure that many of us could recite from memory the parts of the freedoms that I have shown on the left-hand side here, unitalicized. But what I've been thinking about is the part shown in italics, about how we achieve the freedoms, by prevention or rapid diagnosis and treatment, by providing an appropriate environment including shelter and a comfortable resting area, by ensuring conditions and treatment which avoid mental suffering, by ready access to fresh water and a diet to maintain health and vigor, and by providing sufficient space, proper facilities, and company of the animal's own kind. In other words, by providing inputs into the system. And I think, again, this has been very influential in shaping the way that we think about standards. However, as we become more sophisticated in our approach to standards and auditing programs, it's become obvious that providing these inputs does not necessarily result in the kind of welfare outcomes that we want to achieve. And for that reason, I think there's an attempt right now to kind of flip this around and ask, instead of what we should provide, ask what an animal looks like when it's free from pain, injury, or disease, free from discomfort, fear, distress, hunger, and thirst, and free to express its normal behavior. Recently, there have been 3 European projects that have attempted to develop toolkits for on-farm assessment of animal welfare, using primarily animal-based measures. The first is welfare quality. This was a very large EU-wide project with the explicit goal of defining animal-based measures for cattle, pigs, and chickens. That was followed by the AWIN project, which provided additional species coverage by developing protocols for animals that were not covered under welfare quality, for example, sheep and turkeys. and most recently a Harwell, which aimed for a further simplification of animal welfare assessment schemes using animal-based measures, but reducing the time required for the assessment. Importantly, the World Animal Health Organization, the OIE, began the process of setting global standards for animal welfare in 2004, and they now have standards for farmed animals, laboratory animals, working equids, and for some specific situations like transport and slaughter. Recognizing that it would be very difficult to set a global standard due to different capacities, housing systems, and ethical and legislative frameworks in different countries and regions, the OIE chose to emphasize outcome-based standards in their program. And I've put a quotation here. For the OIE animal welfare standards to be applicable globally, they should emphasize favorable outcomes for the animals. Although in some circumstances it may be necessary to recommend specific conditions of the animal's environment and management. So, this leaves open the question of whether we should measure inputs, outcomes, or both, and if so, how do we choose? I'm a big proponent of using outcome-based measures and standards and audits. Most importantly, they're a direct measurement of what we're trying to achieve in terms of improving animal welfare. Another strength is that they identify and address problems that are relevant across situations rather than, for example, being specific to a particular housing system. An example might be something like injuries. We want to minimize those in all housing conditions. Even if their incidence and severity may differ between those situations, we still have as a goal minimizing those problems. As the OIE notes, they allow the exercise of local competencies, but also the professional judgment of people managing the animals in order to resolve problems. And they facilitate continuous improvement, and I'll talk a little bit more about that later on. Overall, outcome-based standards have much more flexibility generally than resource and management-based standards. Another thing I like about them is that they can get us out of having fruitless arguments. I found this picture and this text on a website that is designed to teach science to school children. And the question says, how many angels can dance on the head of a pin? This is a question that has been pondered over the centuries. Can it be answered using the scientific method? In my career, I certainly feel like I have been mired many times in the argument of how many angels can dance on the head of a pin. So, for example, I would get a question from a producer. So you say that hens have to be given 6 inches of feeder space. What if I only have 5.9? Have you ever evaluated 5.9 or 5.8? And the answer is, we'll never have a high degree of scientific certainty about many aspects of animal welfare when we try to be very prescriptive about things. And that really leaves us with 3 courses of action. Either we can stick with our professional and practical judgments and risk that producers, for example, are penalized for fairly minor effects on animal welfare. Or we can retreat into ambiguous language. That is, instead of specifying a particular number, we can say that resources should be adequate or sufficient. And that's a huge source of problems in many standards, that there will be different judgments made by different people about what those words mean. And that's a big source of frustration often in auditing programs. Or we can attempt to find valid outcomes that will answer these kinds of questions. One of the main strengths of outcome-based measures is their use to address problems that are multifactorial. And in the real world, animal welfare problems are often multifactorial. Let's take feather pecking as an example. Feather pecking is an abnormal behavior that can lead to hens in flocks becoming denuded of feathers and can lead to injury. And many factors can affect whether an outbreak of feather pecking occurs in the first place and how severe that outbreak is, including genetics, the quality of the rearing environment, diet, foraging enrichments, light intensity, and poor health status. Rather than trying to specify all of those factors by the use of resource and management-based measures, a more effective approach is to focus on the problem, which is feather pecking, measure it, and then systematically vary the input factors to reduce the problems. This is not to say that the process of using outcome-based standards is straightforward, and I think that's very nicely illustrated by the OIE broiler standard for space. The OIE does not provide a numerical value for the amount of space that's to be provided to broiler chickens. What they do is list a lot of potential outcome-based measures, including things like injury rate, contact dermatitis, behavior, parasitic infestations, and feather condition, as well as the considerations that need to go into selecting and using outcome-based measures, ranging from management capabilities to ambient conditions to the housing system to genetics, biosecurity strategies, and the market age and weight of the birds. Given that complexity, I'd like to walk through the general process that we use when developing outcome-based measures. The first step is to identify the most significant welfare problems, which will be sector-specific, and then determine the possible indicators we can use to measure those problems. During the development phase, we then validate the indicators to assess them for their biological relevance and to create scoring systems, assess how practical it is to use that scoring system to make measurements in situ, and then determine where, when, and how they should be measured. We then, in the next phase, actually measure them in situ. The next phase is to analyze the risk factors for the occurrence of that particular problem. We can certainly gain a lot of information about that from experimental studies, but it's also very important to carry out epidemiological studies to understand how those risk factors interact in situ. And the goal here is to capture as much variation as possible in a real-world setting. The next step is to inform about the incidence and severity of the problem via the process of benchmarking, and I'll talk about that in more detail later on. And the last phase is to provide support for continuous improvement by working to help systematically reduce risk factors, then to measure the outcomes to see if the interventions have been successful, and if not, to return to systematically reducing risk factors and measuring again until we reach our goal. In presenting those steps with arrows, I've probably given the impression that it's a process that we go through, we get to the end, and we're finished. But it's not really like that. It's not one and done. In fact, it's more like a circle. We may find as we go through these different stages that something in one of the stages causes us to have to go back to an earlier stage and reconsider the process. I'll talk more about some scenarios in which that might occur later in the talk, but just in a very broad-based way. For example, we might find when we start measuring the problem in situ and analyzing the risk factors that even though the problem we've identified is a serious animal welfare problem, we really are very constrained in our ability to address that problem using outcome-based measures. To illustrate that point, I'm going to provide some examples from each of 3 areas: develop, inform, and support. And I'll start with develop in terms of validating indicators. This slide shows 3 animal-based scoring systems that are in use for welfare assessment of broilers. The top panel shows the scoring for foot pad dermatitis, the middle panel for hock burns, and the lower panel for plumage cleanliness. Certainly, foot pad dermatitis and hock burns can be problems in their own right, causing pain to the birds. But all three of these scoring systems are also proxy measures for litter condition and potential problems with lameness. However, looking at these scoring systems raises some questions. Do scores really discriminate between good and bad outcomes? That is, are they biologically relevant? So as humans, we are able to discriminate, for example, between a score of 0 and a score of 1 on foot pad dermatitis. But does that difference really make a difference for the bird? And I think the answer to that question is very tied into the question of what are our practical and ethical goals for the scoring system. Looking at the bottom panel, for example, if our interest is in distinguishing between clean birds and dirty birds, where do we draw the dividing line? What level of dirtiness indicates an unacceptable level of litter condition or an unacceptable problem with lameness? In general, for scoring systems, simplicity is better than complexity in terms of its practicality for use in on-farm assessment. A key question that we need to address then is how we simplify scoring systems without losing critical information. My next example comes from the area of informing in terms of setting benchmarks and tolerance limits. This slide shows data taken from an epidemiological study on dairy farms looking at the incidence of lameness. The x-axis shows the percentage of lame cows and the y-axis the percentage of farms. So a few things to notice about the graph. The typical incidence of lameness on about half of dairy farms was 30% of cows. Fortunately, there were no farms that had all lame cows, but there were also no farms that had no lame cows. The best farms had about 10% lame cows. This slide illustrates for me something very important about the process of benchmarking and establishing tolerance limits for a set of standards or an audit, which is that the process is inherently subjective. And when I talk to producers now, what I tell them is that they should decide in advance very clearly what their programmatic goals are. Is the goal of establishing the tolerance limits to move all of the farmers who are in the yellow highlighted box here to the level of the typical dairy farm? Is it to move the typical dairy farm incidence down to the level of the best producers? So again, this is a value judgment. It's not really a scientific judgment. As scientists, we can determine these percentages, but it is then an issue of programmatic goals to decide where to set the tolerance limits for a particular program. There are a number of other complexities associated with benchmarking. What if the problem is important but rare? In that case, the sample size that we need may be too large to include the measure in our assessment. As an example, bumblefoot in hens is an infectious foot disorder. It's very painful. It is an animal welfare problem, but it appears to be very rare, occurring in fewer than 1% of hens in a flock. We'd have to sample a very large number of hens in order to determine on-farm incidence, and then there's very little room left for creating tolerance limits because of the rarity of the problem. And for some of these rare problems, we may then either need to take them out of the standards and the auditing, or rely instead on record keeping and documented compliance with protocols. An example would be something like non-ambulatory dairy cattle, which we hope occurs very infrequently, but when it does occur, it's an important problem that has to be documented with appropriate corrective actions applied. Another question is, what if it's not one benchmark, but many benchmarks? This is something that I'm really struggling with right now. I'm working a lot with the laying hen sector, both from the industry perspective and also from animal welfare certification programs. And it's becoming very obvious that many of the important animal welfare outcomes are going to be affected by factors like age, production stage, genetics, region, and housing system. And that's going to make the process of setting benchmarks very, very complicated. The last area I'd like to address relates to support, and particularly how we systematically reduce risk factors and encourage continuous improvement. An important question to ask when we include an outcome-based measure in a standard or an audit is whether or not there are clear corrective actions to reduce or eliminate the problems. If not, including those measures can be very demoralizing and counterproductive. And we still have many multifactorial problems that we are disentangling. An example of that might be fearfulness, which is certainly important. It's one of the 5 freedoms. But at this point, we still have a very poor understanding of some of the causes of fearfulness and how the problem can be corrected. In those cases, we may need to transparently make a best guess about input-based measures that will help in reducing the problem. I'd like to return to the discussion about how we choose what kinds of measures to make. I'm going to make some suggestions about situations in which resource management or outcome-based measures should be used. These are by no means comprehensive, but they do represent things that Cass and I have been discussing about the ways to take a more systematic approach to making these kinds of decisions. So when should we use resource-based requirements? All of the standards that I'm familiar with use resource-based requirements for things that are required for health and physical condition needs. For example, food, water, and preventative health care. For example, vaccinations. It also makes sense to use these kinds of requirements when the resources are required to satisfy behavioral needs or wants, although that's certainly more variable now between different existing guidelines depending on goals. I will note, though, that in both cases it may be very challenging and uncertain to specify exactly how those resources should be provided. Another situation to use resource-based requirements is when the outcome is difficult or impossible to measure in a practical setting. Dehydration would be an example. We, in most cases, don't have adequate measures of dehydration in situ, although we can measure it in a laboratory. So in this case, we would want to be very prescriptive about how water is provided. And another example would be complex behaviors. These are very time-consuming to measure, and they require a very high standard of training. A final situation is when there is a high level of scientific certainty that the resource and/or the quality or quantity of the resource will actually improve welfare. Situations in which we should use management-based requirements, including for prohibited or restricted practices, that is, practices that are restricted in terms of how and/or when they can be performed. Examples would be things like mutilations, tail docking, beak trimming, euthanasia in terms of conduct of permissible methods or methods of killing that are not allowed. An additional situation is for important inputs that cannot normally be measured by outcomes. We can think of these as preventative measures. For example, emergency management plans, the veterinary client-patient relationship. We certainly hope that events do not occur that lead to an emergency that results in the injury of animals on farm, or that there are not serious health conditions, but we use And the last is for point-in-time events that cannot be audited later. An example of that would be something like transport from one facility to another. When should we use outcome-based measures? We've already discussed their importance for addressing multifactorial problems. There are also opportunities for the use of outcome-based measures by refocusing our questions on what exactly the problems are that we're trying to solve. I mentioned earlier all these discussions I've had about feeder space for laying hens. Does it need to be 6 inches? Can it be 5.8 inches? And the truth is that we have very little scientific certainty about exactly how feed should be provided to hens, particularly in more complex systems like cage-free systems. Going back to asking what the problem is we're trying to solve, and in fact back to the 5 freedoms, we are trying to solve the problem of providing the diet in a way that provides ready access and maintains health and vigor. So in other words, we want healthy, normal hens that are not in a situation where they have to aggressively compete for feed. And we actually can measure those outcomes. At the flock level, for example, we can look at body weight, egg production, and mortality. And I know these kinds of flock and herd level measures are criticized because they don't focus on the individual animal in the system. But we can also look at that, at least a sample of that, by looking at uniformity in body weight among the hens in the flock and looking at the body condition of a selected number of individual birds. And fortunately, we also have some very good proxies for aggressive competition in terms of looking at comb wounding and head feather loss. However, the 2 situations we've been discussing, the first in which the outcome itself is the problem, and the second in which the outcome is replacing input measures, may lead us in very different directions when we think about corrective actions. Let's start first with the example of the situation where the outcome is the problem, in this case, feather pecking. We can measure the extent of feather pecking in the flock, and because of research conducted at the University of Bristol, we know that there are about 40 potential changes that producers can make in their housing or management to reduce the incidence of this problem. So those changes can be applied systematically and as they are relevant for that particular management and housing system. The situation becomes more complex, I think, when we are talking about outcomes that replace inputs. Taking feeder space as an example, there is not a one-to-one relationship between feeder space and the outcome-based measures that we are using to assess feeder space. In other words, we can envision other inputs that would also affect these particular outcomes. For example, there could be a health problem in the flock, undiagnosed, that is having effects on production variables, body weight, and contributing to problems with aggression in the flock. In this case, the corrective actions will involve determining the actual causes of the problem rather than applying a systematic strategy of reduction for a particular problem. A final but important point I'd like to make is that this really requires us to rethink our approach to auditing. For continuous improvement, we need a commitment to revising and refining standards and benchmarks regularly. Many of the measurements of interest probably cannot be made by the auditors. It's very time-consuming to collect some of these measurements. They may involve handling the animals, and importantly, it may be a matter of historical recording rather than recording during the brief period of time that the auditor is actually on site. That means the measurements are going to need to be made in real time by the people who are at the site regularly. In this sense, the auditors become record reviewers. In addition, auditors will need to determine what is an acceptable approach to corrective actions and what is not an acceptable approach. And that's a very different task than coming to a site, evaluating how well the standards are met, and then deciding if a facility passes or fails. This also raises issues related to transparency. When we take this kind of approach, there's a potentially long time frame for correction of problems. Patience is required. But how will that be viewed by the public? Is this going to be sufficient for risk management and assurance about animal welfare? It's very important for us to have the answers to those questions in order to facilitate the widespread adoption of these measures. I can take questions.
Thanks, Joy. If you'd like to turn your camera back on, we can ask the questions.
Yes.
Great, thank you. There were a number of comments in the questions congratulating both you and the early career researchers on your medals, so I'll just pass those along first. Congratulations from me as well. The first question from Berta is, to what extent will precision livestock farming technologies be useful for the assessment of animal welfare at farm level?
Thank you, Berta. I think that's an excellent question. I think that is going to result in huge improvements, obviously, in our ability to collect and record this information in real time. We have a variety of outcomes now, probably, that we could collect in that way, and as those technologies improve, we'll have more that we can collect. So I think that's going to be a big advancement in terms of at least collecting and getting the information necessary to benchmark these kinds of measures.
Thank you. Please do, the audience, please do put your questions in the question box. I'll see if anything else. While I'm waiting, you did mention well towards the beginning the importance of training the auditors. Do you have any experience with that?
I do have a little bit of experience. Cassandra actually has more experience than I do. She works regularly with PACO, which is an auditor training organization here, but I have a little bit of experience doing that, yes. Just in a classroom setting rather than in an on-farm setting.
Thanks. So we're waiting for some questions to come through. And say, but I like your focus on the animal-based measures rather than anything as one of the main as the outcomes. I strain myself very well. I like your focus on the animal-based measures and outcomes. I've seen some welfare scoring systems recently where it's been very much on the environmental side, factor side, rather than the animals, and I think it's a shame.
Yes, I think the environmental side in some ways is a little bit easier. As I mentioned, I'm working now with the laying-hen sector, and we really do have these complexities of benchmarks are not just a moving continuous improvement situation, but we have all these complexities in terms of when they should be measured, how they're going to change over time. And an issue I didn't really have a chance to discuss was the additional complexity that's involved in benchmarking when you have not only an incidence of a problem, but a severity of the problem in the scoring system. That adds another layer of assessment about what you're trying to do first. Are you trying to decrease severity? Are you trying to decrease incidence? Are you trying to do both things at the same time? So we have a lot of work ahead of us, I think, to put these into practice.
Nice to see it then. And we have got a few questions coming in through now, thanks. If I say, just scroll back up my list. So Lucy Aldermass, is the question of what are we aiming for mainly an ethical one, i.e. what standards will the consumer accept, or can it be simplified to what is a meaningful improvement on the current condition?
Well, yes, it's certainly partially an ethical question. I mean, there is a scientific element of that in terms of, let's say we're talking about a scoring system. From a scientific perspective, we are aiming to make that scoring system as biologically relevant as possible. And I think that's a scientific issue. But from the benchmarking perspective, that is going to vary from one program to another depending on their audit rules. So if you're an animal welfare labeling program, for example, like the RSPCA, you might decide programmatically that you only want producers on your program that have the best outcome-based metrics. Whereas if you're an industry-wide program, like some of the programs that I'm working with right now in North America, you might decide that your goal is to create an overall improvement within the industry as at least your first pass. But again, the nice thing about these kinds of metrics is that you can revisit that goal later on as the incidence and severity of problems actually change.
Okay, so thank you. I've got a question from Georgia Mason. Are stereotypical behaviours used much in audit schemes? She says she gets a feeling not, but she doesn't understand why.
I'm sorry, I didn't understand the question. Could you repeat that?
Are stereotypical behaviours used much in audit schemes?
No, not for farm animals. I think the problem is the issue related to actually measuring those on farms in order to audit them. If I understand your question, Georgia. If we have proxies for those kinds of behaviors, for example, feather pecking as a repetitive behavior, then we're able to audit them. But otherwise, they are, difficult because of the behavioral observations that are required. So certainly on farms, they're not, although there are examples of that kind of analysis being conducted in zoos, for example, with elephants.
Thank you. Stephanie Wisdom asks, from your experience, do you have any tips on how to encourage livestock industries and producers to adopt more robust livestock auditing standards?
Well, again, I think this is very much an evolutionary process. I think we have seen over time in the United States, at least, that the standards become more stringent as more and more producers are involved in these programs, and as they have an understanding of how they can correct actions on their farms or correct problems on their farms. And again, I think that's going to be one of the strengths of outcome-based measures. I will say there is some nervousness about kind of when do the benchmarks stop moving? when is a corrective action finally a corrective action? And so that's an issue that we're dealing with now in terms of discussions about incorporating these kinds of measures into standards and auditing programs.
So getting quite a few questions through now. Catalina Diaz-Vasco asks, how do farmers react to the change in focus between input and output measures?
Yeah, I think I just answered that question a little bit. I think they're generally positive about outcome-based measures because they see that it will allow them to use their professional judgment and to consider the circumstances on their own particular farm, their management capabilities, their resources, what they're actually willing to devote to correcting problems. So that's a very positive for them. The main concerns, I think, again, are if the benchmarks keep moving. So let me give an example. I'm passing my audit right now because the auditor can come in and measure all these things and they're all fine and I pass. What happens if the benchmarks keep moving and I develop a problem that I can't pass my audit anymore? So that's a very practical consideration for them, particularly if the audit outcome in terms of passing or failing is actually linked to them being able to sell their products into a particular supply chain. So that becomes a big economic issue.
Yeah, we've got time for at least one more question. Lindsay Goodall asks, voluntary versus obligatory programs, is there any differences in your recommendations?
I'm not sure what's, by obligatory, you mean something like a legal standard or a retailer program, or?
I think so. So I don't know, not sure about farm animals, but certainly in laboratory animals in the UK, we've got the Home Office in terms of regulations and so on.
Well, I think the general principles apply to both, although I think it's much simpler if we're talking about a regulatory, a legal program versus a voluntary program. It's much more difficult to change legislation. And that makes it harder to include continuous improvement strategies. It's not impossible, but it's more difficult.
Thank you. And I think we've got time for one final question. Susan Millman asks, she agrees with the value of epidemiological analysis to help with the benchmarking risk factors, but these rely on reliable databases. There's real challenges there with, sorry, my question's just disappeared.
Oh, okay. Yeah, you also cut out a little bit. Your mic cut out a little bit.
Yeah,
so.
sorry, I'll just see if I can find back to the question. Okay, I've got it. So she agrees with the value of the epidemiological analysis, but benchmark for help with benchmarking risk factors, but the analysis relies on having reliable databases to work with. And there's some real challenges there working with proprietary information and logistics. Do you know if anyone is working on the technological aspects of that? So getting relevant animal-based outcomes into some of the existing software programs farmers use?
I don't, sorry.
Okay,
no.
I think that's everything. Thank you very much.
Okay, thank you.
Okay, so I'm just so the next talk will be from Erin Ryan of the University of British Columbia in Canada.
Hi everyone, and thank you for joining me today for the presentation. And yes, I'll be talking about a new topic in humane rodent control. So generally when I'm talking about rodents, there's many species worldwide and even in North America where I'm based. But when I'm talking about rodents, these are the three that I'm talking about mostly. These are the commensal rodents. So up top, the house mouse, Norway rat, and roof rat. And I think this topic's really important because whether you realize it or not, you are a customer of pest control. And it's important for a number of different reasons. And it starts with health and safety of both people, and as somebody who also works for an organization that shelters animals, it's important for the safety of the pets that we house and care for as well. It's important for property protection because rodents can damage structures with gnawing, but they can also chew on things like wires, which makes them a fire risk. And of course, it's also important worldwide for conservation because many animals that grew up without natural predators, rodents can suddenly become a very voracious predator. Rodenticide is the most commonly used tool to control mice and rats around the globe. And we know that it's not a humane alternative. We know that mice and rats suffer for days or even weeks as the rodenticide takes effect, but we also know that it's a serious risk to other animals. So for example, coyotes, hawks, eagles, owls, other raptors, any animal that might eat poison carcasses can also experience secondary poisoning where the poison builds up and they experience the poisoning themselves. And from my work, I'm also learning that there's a lot of impacts that we don't yet know the full scope of. So for example, rodenticide appearing in suspended particulate matter in water systems where fish live. And we don't know yet the full impacts of rodenticides in our water ecosystems. And when it comes to rodent control, a lot of my background is in urban wildlife control, and I felt like I had humane alternatives to suggest for most species. But when it came to rodent control, I felt like I was helping people choose between kind of the least worst options. So snap traps, pictured here, can produce a relatively humane death if they're placed appropriately. But if they're just left outside, they can they can capture other species like birds and other small animals. And the performance of these traps varies extremely widely. So you need appropriate spring power for these traps to be humane, but there's no, at least in Canada, there's no regulation saying how powerful that has to be or which traps are better than others. Glue boards are an unfortunate tool as well. Most often trapped rodents die from suffocation when they get their face stuck in the glue, but also times from dehydration or exposure after just being trapped on the glue board for a number of hours or even days. Live trapping is still an option in some limited circumstances, but this is very intensive in terms of manual labor and not particularly effective if you're dealing with an infestation level. There is budding research coming into fertility control. There's a product that is available in the United States right now, but it's not commercially available in Canada or elsewhere in the world that I'm aware of. But it is an interesting tool to keep an eye on. So the trap that I'll be walking you through today, or the trap that I did my research with, that was the Good Nature A24 rat and stoat trap. So this is a captive bolt trap that was developed in New Zealand and is starting to make its way through North America and the rest of the world. So you can see here, this is kind of an expanded view of the trap. But basically this side would be mounted to a tree or a plank of wood or something secure that you can put the stand to. You'd have your lure attractant, so that could be your own special blend, or it could be purchased from the manufacturer. And then there is a pressurized bolt using a small carbon dioxide canister. And the idea here is that, the rat smells the tasty lure up top, they move aside this trigger needle, and then that pressurizes the bolts, which ideally instantaneously crushes the skull. And a convenient part about these traps is that they actually automatically reset. So once the trap fires, the body drops out the bottom and the trap is ready to go again. Each canister will do about 26 kills. And then of course the trap's ready to go again, and any bodies that are scavenged from the bottom of the traps are poison-free and safe for any predators. So for my research, for phase 1, I was really interested in knowing the non-target interactions. So what types of animals we're visiting and was it safe for them? Because of course our animal ecosystem is very different in North America, in Canada, than it is in New Zealand. And then phase 2, I wanted to look at a humaneness evaluation and seeing, was this trap actually as humane as it was marketed to be? So in this phase 1, what I wanted to examine was the effect of the squirrel blocker attachment. So you can see that here. So basically I used that as a treatment group. So half the traps, half the time were fitted with one of these squirrel blockers and the other half they weren't. And for this phase of the study, I deactivated the traps. So they were baited, but the CO2 canister was not in place. So it was essentially harmless. And each trap was fitted with a motion activated video camera so that I could watch each of the species interacting. And I saw in the beginning a lot of what I expected to see. I saw different species of mice and rats. I did see squirrels and raccoons and skunks trying to access the trap, but a couple other small numbers of rodents. But what I wasn't expecting to see was 23 different species of birds. And then keep in mind, the field of view on these cameras is quite narrow, and these traps are placed specifically in areas that I expected rodents to be. And still, there was quite a few bird visitors. And in terms of overall species recorded during the project, birds undoubtedly accounted for the majority, but they didn't necessarily interact with the trap the most. So you can see here, really, although a lot of birds visited the traps, the majority of them, whether or not there was a blocker, had no interaction whatsoever. They simply investigated the trap, or they touched the trap, or in birds' cases, that meant they perched on it. They love to perch on it. But in terms of actually getting involved with the trap, there was very little instances. And essentially what this boils down to is I was curious about how many animals were involved with dangerous interactions. So animals that actually tried to insert limbs into the traps or actually tried to enter the traps with and without the blockers. So you can see that without the blocker, we did have one bird enter the trap. We had 9 raccoons, 27 squirrels, and 14 rodents. Versus with the squirrel blocker attachment, it effectively removed the risk to all of the non-target animals, but mice and rats were still effectively able to enter the trap. And you can see here, this is just an example, raccoon sticking their paw right up the trap when there's no blocker in place, and squirrel actually sticking their head right into the body of the trap with no hesitation whatsoever. So from that first phase of the study, I was surprised by the high biodiversity and a large number of species that could potentially be exposed to any rodent control devices, whether that's the good nature trap or rodenticides or snap traps. But in general, the good nature trap presented fairly low risk to birds, and the use of blocker very effectively removed the risk to non-target animals. And at the same time, we felt that the blocker didn't inhibit rodents' ability to enter the trap. And for phase 2, it's essentially the same setup, except that I did have the gas canister in place, and all traps were fitted with the blockers to make sure that we weren't presenting any risk to animals like squirrels and raccoons. And I wanted to show you a sample of how the trap works in action. So I'll show you the video. It's not bloody or gory or particularly upsetting, but if you want to look away, feel free to do so. I'll let you know when I'm back away from the video and it's safe to look again. So you can see the mouse is going up into the body of the trap. They kind of fumble around a little bit. They're entering inside. And once they move aside the trigger needle, the trap fires and the body drops at the bottom. And that's it. That's all there is to it. The body drops to the bottom and they were not recorded moving again on camera. Okay, we're away from the video, but we are looking at X-ray if anybody's sensitive to that. So I wasn't able to recover all of the bodies because a lot of the activity, of course, with rodents happens overnight and not if there are people present. But I did recover some of the bodies, which we subjected to a micro CT scan. And this is just one sample. This part of the study is still ongoing, but you can see here, there's a very, very clear break in the spine of this mouse. And ideally, the trap should be hitting the skull, but that would be for larger animals like rats. So we are looking through the team to see, how effective that break would be in causing instantaneous death. And you can see here that these are kind of all of the interactions with the good nature trap. So when we were just looking at mice and rats, and we recorded a large number of mice and rats, a lot of them that had no interaction, unfortunately. But there was quite a few that investigated the trap. And you can see with mice, they were much more willing than rats to enter the trap. But unfortunately, it means that we got much more mice that triggered the trap than we did any other animals. And unfortunately, even though the blockers were effective in the first phase for excluding squirrels, we did have 2 squirrels in the second phase of the study that were still able to trigger the trap. So, essentially what it boils down to is that the traps were very frequently visited, but very few animals actually entered the trap. So, although we hope to collect a large number of observations on the kills, only 10 kills have been observed to date, and that was 5 house mice, 3 deer mice, and unfortunately the 1 gray squirrel. In general, rats were very unwilling to enter the trap, no matter what type of bait we tried to throw at them. And again, although the blockers were effective in study 1 for squirrels, we did strike 2 squirrels in the second half of the study. Our observations showed immediate or near immediate cessation of movement and apparently instantaneous death in mice, but we don't have sufficient data to make any conclusions for rats, unfortunately. The analysis for the study is still ongoing, including the micro CT analysis. And in short, I believe that it's still a very, very promising tool, but it's not a substitute for a good integrated pest management strategy. And I feel like a lot of the rodent problems that I witnessed at these 2 sites could have easily been mitigated by some cleanliness, sanitation, and exclusion measures. I just want to take a minute to acknowledge my supervisors from UBC, Canadian Wildlife Service, who donated the supplies that I used for the project, and my funders, the Okanagan Indian Band and the Aboriginal Graduate Fellowship. Thank you, and I'm looking forward to taking any questions.
Thanks, Erin. Do you want to turn your camera on? Thank you. So first question is from Stephen, asking what's the bait you used? You did mention using several different ones with the rats.
Sorry, what's being used for?
For the bait.
Oh, so it was a combination of things. So I did use Good Nature's manufactured, a non-toxic chocolate lure. But in the second half of the study, where I was trying to maximize trapping efficiency, I was also using a combination of peanut butter and Nutella. And I'm just pulling up those graphs now as part of the research, and it is seeming that just peanut butter and Nutella seem to be the most effective option for mice. But the type of lure didn't really affect rats and other rodents.
Hugh Gollage asks, is the trap to be shown to be more effective in killing rodents in New Zealand?
You know, I wondered about that too, because there have been some studies looking at animals that are in more urban spaces that are kind of more used to tools that might cause them harm, or to environmental man-made things that might cause them harm, tend to be more wary. So rats that are naive to pest control are more vulnerable to forms of pest control. So it is quite possible that in a more, rural setting, the more naive rats are more vulnerable versus, we're looking at these city settings where they're living alongside us all the time. It could be that they're much more wary of man-made things.
Follow up from that one from Jamie. He did say, given the low kill rate, how effective do you think they would be if you absolutely blanketed an area of traps? Would there be a ceiling due to an individual's just being a bit too trap shy? I think you've probably just answered that with that.
Yes, but I also think what I would be curious to know, there's not really any data on how trap shy animals are, for example, for rodenticide bait or snap traps. So I would be really curious to see somebody set up, for example, a video camera on a bait box and see how many mice and rats pass a snap trap before one actually gets inside.
A few things on the bait. Just to pass on to you that from Sam Watson says that he can confirm that, or they can confirm that their house mice adore Nutella and chocolate coated raisins. And thank you for the valuable work. They look forward to hearing more about it in future. For a follow up, the bait, do different baits attract different animals?
The bait does attract other animals, squirrels and raccoons. During that first phase of the study where the traps weren't set but they were baited, the raccoons and squirrels, no hesitation, would put paws up in the trap or stick their heads in the trap. So you do have to be careful with that, because of course any bait that's made to be attractive to mice and rats is going to be attractive to any other scavengers.
You mentioned that there's approximately, so Elizabeth Carter asks, you mentioned that there were approximately 26 kills per canister. Is each kill is each kill equally effective, and does the canister run low towards the end in terms of effectiveness?
So my understanding is that it's 26 full pressurized kills, and then after that is when you start seeing kind of the deterioration. So it's recommended you change it after 26. But there is also, I think they also recommend a 6-month change just in case. So if you haven't hit your 26 by the end of 6 months, then it's recommended that you change out the gas canister to make sure.
Thanks. Mustafa Fargal asks, what is the estimated cost of the trap?
So it depends where you're getting it from, because when I initially started working with these traps, they were only available in New Zealand, so I had to order them from New Zealand and pay, the freighting charges. There is a distributor in North America now, so the traps are running, I think for a full kit, you're looking at about $129 base rate, but then any shipping and duties you have to look at. So it is a more expensive investment upfront. But once you have the traps and the bodies, then the only thing you're ordering is the lure, if you're purchasing the lure versus making your own, and the gas canisters, which are relatively affordable.
Thanks. Still got plenty of time still. Shauna Laven asks, what is the fertility control you mentioned that's available in the US?
So the name of that product is called Contrapest. So I have been looking into it. It's unfortunately not available in Canada, which I would love to see somebody do some research on that. But the idea is that it inhibits fertility. So it produces, fewer litters and smaller litters. So it works on both the males and the females, basically acting on the follicle stimulation in the females, but in the males also inhibiting the generation of sperm.
Tom Smulders asks, would it make more sense to use the traps inside houses rather than garden green areas so that you get fewer side kills and target the rodents that are the real problem inside the house?
Yeah, I think one of the things that I'm concluding out of this is if you're using these traps outdoors, I would recommend always using the squirrel blocker because you never know who can get inside. But if you're looking at inside a house where you have mice, you have rats, you could easily set up the traps in wall voids or behind appliances, areas that you specifically expect mice and rats. And I think that might help with trapping efficacy indoors.
George and Mason asks, what scope do you think there is to use it for lab mice? It would be nice to have a practical alternative to aversive CO2.
Sorry, please, my fellow pest control enthusiast, Wesley. So I know there's been interest in the laboratory setting because, we know there's such strong aversion to CO2 and other inhalant gases. And a physical method that wasn't so demanding on technicians is desirable. There is some colleagues at UBC that are looking at this, but I think the trouble that they're finding is that, even though it's a lot of the same species of rat, they tend to behave differently in the wild versus the lab setting. And so the tricky part, at least they've been finding in the lab, is finding that balance between training them versus not training them. Because if they're naive to the trap, it'll take them forever and ever before they'll stick their head in far enough to activate the trigger needle. But if you train them on the trap when it's inactive too much, they also learn how to sidestep the trigger needle because they don't like having their whiskers or their nose touching things. So it's a fine balance there. I think it might still have potential, but those are some of the early challenges that we found there.
Thanks. I think I have one final question. Do you know what the percentage of the carcasses were that were scavenged?
Sorry, how many?
How many of the what percentage portion of the carcasses were scavenged by other animals?
So of the 11 kills, I only recovered 4 bodies. So I did see a couple of them be scavenged on camera, but some of them, fell kind of below the line of sight. So I didn't see anything come get it, but I was checking every day trying to recover. Yeah, so I managed to recover 4, but it looks like the other ones were all scavenged.
Right, thank you very much. I think we can move on to the next presentations now.
Thank you.
Okay, so this final session is the speed bits of talks, the speed presentations. All 5 presentations are going to be shown back to back. Please could you ask questions throughout, and it'd help if you could mention who the questions for when you ask them, and we'll ask all the questions at the end. The final thing, could the speakers be the 5 speakers be ready to turn their cameras on at the end of the 5 presentations, please?
Do you think that current practices of pup counting in breeding facilities provide accurate numbers of pups born and dead? Perinatal mortality is a major welfare and economic issue in laboratory mouse. Perinatal mortality in lab mouse is characterized by the death of newborns within the first 3 days of life and is often followed by the disappearance of the dead body in the next minutes or hours due to cannibalism. So it makes pup counting and tracking very challenging. In this study, we wondered whether daily cage check, which is a common method used in mouse breeding, is a reliable counting method to estimate litter size at birth and mortality. To address this question, we compared 2 counting methods. We used a standard method called daily check counting, where cages were daily checked until pup detection on day 0, and pups were counted once detected. We compared this method with an experimental method called video track counting, and where daily cage checks and pup counting were accompanied by video analysis to detect the cannibalism events that happened between start of parturition and next cage check. Litter size estimation was based on the number of pups born and found dead at the first detection on day 0 for both methods, but it also included the number of pups cannibalized between birth and day 0 for video track cages. There were 2 social configurations, since litters can born in trio cages with 1 male and 2 females, with the presence of another litter or not. Let me show you the main result of pups count comparison between both counting methods. First, you can see that we did not find any difference on the numbers of weaned pups per litter between both methods. This result is very important because it indicates that our video track counting method did not increase the risk of pup loss compared to the standard daily check counting method. However, you can see that the estimates of pups born and dead per litter were higher when daily check were combined with video analysis compared to daily check only. Hence, litter size and mortality were underestimated even when cages were daily checked and pups counted once detected for the first time. To understand whether cannibalism could play a role in the discrepancy between those results, we explored data from video track counting and calculated the percentage of pups that were already cannibalized before the cage check on day 0. Here we go. This figure shows the percentage of pups alive, found dead, and already cannibalized at the first cage checking on day 0, according to the social configuration, since we had a strong impact of this factor. On the left, you have trios without the presence of another litter, and on the right, trios with the presence of another litter. You can see that a high proportion of pups were already dead and cannibalized between birth and the next cage check on day 0, and it concerned until 25% of the litter in trios with the presence of another litter. In terms of percentage of dead pups, 52 to 69% of the dead pups were already cannibalized before this cage check, depending on the social configuration. You have to remember that those pups could never be detected without video analysis. In conclusion, cannibalism clearly masks perinatal mortality around parturition, and this phenomenon is aggravated when another litter is present in the cage. Now, we are wondering whether this problem concerns a small proportion of breeding facilities or if it is a widespread issue.
Hi everyone, my name is Katie Cronin and I lead the Animal Welfare Science program at Lincoln Park Zoo in Chicago. Today, I'm going to talk about how valuable animal space use patterns can be for gaining insights to welfare. Generally speaking, animals will spend more time in locations where they feel comfortable and avoid areas where they feel less comfortable. We rely on this general truth all the time, for example in preference tests when we ask animals where they want to spend their time. By now, we are all familiar with the idea that positive affective states are associated with positive animal welfare. So it follows that we can use space use patterns to draw inferences about aspects of their environment that promote good or bad welfare. About 5 years ago, Lincoln Park Zoo developed and released an app called Zoo Monitor for recording animal behavior and space use that's being used by hundreds of institutions around the world. At Lincoln Park Zoo, volunteers who have established inter-observer reliability are using the app to collect space use data that has proven especially valuable for evaluating and enhancing welfare, and now I'll share a few examples. First, we relied on space use data to gain insights into whether chickens were comfortable in a new animal visitor interaction program. We had redesigned all of our programs so that animals had choice, and we wanted to measure whether chickens were comfortable engaging in this program. Here you can see a top-down view of the chicken habitat, and the little gold boxes along the bottom show areas where people could approach and feed chickens through the mesh. Chickens had food available in their enclosure at all times as well. Space use is displayed as a heat map, and the red areas indicate the areas of most frequent use. This plot gives us confidence that visitors were not inducing a negative affective state in the chickens that was expressed by chickens avoiding the area near the visitors. The next example comes from takins, large hooved animals native to Tibet. We considered takin space use patterns alongside data we were collecting on shade availability in outdoor habitats throughout the zoo, and we found that in warmer temperatures, takins were disproportionately using shaded areas of their habitat. We then used these data to make recommendations about where to place a new shade structure. We considered how toucans use space in lower temperatures to inform placement of a shade structure that essentially increased the amount of preferred habitat in sunny conditions that was available to the toucans. Finally, we compared the space use of 5 species of primates in the presence and in the complete absence of zoo visitors to better understand the visitor effect on primates. This panel shows a box plot for each species, and you can see that the medians within each species do not differ between conditions when visitors are present and visitors are absent. This space use data gives us confidence that at least for these individual animals in these habitats, animals are not adjusting their space use patterns to avoid visitors. I hope I've shown you the value of considering space use for gaining insights to welfare. There's more information available on the supplemental PDF. Thank you for your attention, and I look forward to answering questions.
Hi everyone, my name is Vicky Neville and I'm a postdoc at the University of Bristol, and I'll very briefly summarise a study that we conducted last year to assess whether pet rats could inform laboratory rat welfare. So I don't think that I need to state the huge importance of studying laboratory rat welfare, so I'll jump straight into explaining why we decided to study pet rats. And one real issue in studies of laboratory animal welfare and animal studies more generally is that high levels of standardization, for example, in terms of environmental conditions, can mean that results aren't generalizable. But pet rats and their environment vary in a number of ways across owners, which allows for greater heterogeneity. Also, as animal welfare scientists, we're often stuck in the really tricky predicament of wanting to do our best for animals, but then studying their welfare often means using animals in research and sometimes inflicting conditions that we think are less than optimal. So using pet rats obviates the need for this because we can capitalize on heterogeneity in conditions to investigate potential welfare issues, and no rats have to be bred directly for the purpose of the research. With that in mind, we wanted to know whether studying pet rats could provide a useful way to study laboratory rat welfare. So really, we wanted to see if data from pet rats were sufficiently consistent with data from laboratory rats, and we also wanted to take a largely exploratory approach to see whether variables that are considered to influence welfare alter variables that are considered to provide a measure of welfare. So to do this, we conducted a survey of pet rat owners in the UK, which was distributed via social media. We then looked at whether and how predatory exposure via co-ownership of cats, dogs, snakes and ferrets, human interaction and enrichment were associated with health and the reported frequency of a range of behaviours, given that these variables have all been linked to welfare in some way. We had a great response to the survey, with 667 owners completing the survey in full. And importantly, we identified a number of well-established and intuitive findings that support the validity of this approach, such as greater play in younger animals, greater health issues in older animals, and greater human-directed aggression in animals who had less interaction with humans. So there are lots of results that aren't included here in the interest of time, so I'm going to stick to the key results. When we looked just at the reported frequency of each behaviour, it was really clear that rats would commonly perform behaviours that are highly restricted in laboratory conditions, like bounding, climbing, digging, food hoarding, and nesting. So finding ways to allow rats to perform these may be important to their welfare. Our study also revealed several behaviours that were less frequently observed when predator exposure was greater, such as bruxing, which was moderately associated with boggling, and you can see both behaviours in the top video, bounding, which you can see in the bottom video, digging, pinning, which is a core component of play, and gnawing. And these are largely consistent with current interpretations of these behaviours in either rats or other species. So it may be that we can use these as potential spontaneous behavioural indicators of welfare. Thanks very much for listening, and please do get in touch with any questions.
I'm Sarah Huskisson with Lincoln Park Zoo. I will briefly be describing pilot and follow-up work looking at the effect of zoo visitors on the cognitive performance and well-being of Japanese macaques. At Lincoln Park Zoo, monkeys work on touchscreens in view of the public. We wanted to assess how visitors influence the monkey's performance on a match-to-sample memory task, as well as the potential welfare impacts of visitor presence during cognitive testing. In our pilot study, we tested 4 monkeys over the course of 6 months. During the testing sessions, we repeatedly counted the number of visitors watching cognitive research. We compared the monkeys' accuracy and response latencies over time and with crowd size to assess learning and potential visitor effects. We predicted that the monkeys' accuracy would increase over the course of the study, reflecting their learning of the task. We also predicted that increasing crowd size would cause decreased accuracy and increased response latencies, suggesting distraction on the monkey's part. In the first half of the study, the macaque's accuracy was at chance levels, and accuracy did not differ by crowd size. Accuracy improved in the second half of the study and was significantly higher when 20 or fewer visitors were present. Response latencies were much faster in the second half of the study and were mediated by crowd size, but there was no evidence of a response slowing effect. Overall, we found the monkeys remained engaged with and learned the task, with some evidence of visitor influence. Since our pilot work did not investigate the impact of other variables besides visitor number, we took advantage of zoo closures due to COVID-19 to tease apart whether visitor presence, visitor number, or other environmental variables impacted the macaque's performance. We tested 9 monkeys. Data collection was split up according to the zoo closure in January and February of this year and subsequent opening in March. For every hour-long test session, we recorded sound levels. When the zoo was open, we also recorded visitor number and location, both on days when monkeys participated in testing and on non-testing days, as can be seen on the heat maps. We found no difference in the macaque's task accuracy between zoo closure and opening. However, prior experience with the task is a better predictor of success than visitor-mediated variables. Response latencies were quicker when the zoo was open, which is likely due to the monkeys' increased familiarity with the task, but response latencies varied across visitor number. Based on this information, the evidence suggests that the monkeys were not greatly impacted by rehabituating to visitors after COVID-19 closures. More broadly, we believe that we are meeting research and animal welfare goals simultaneously. We encourage others to adopt more detailed monitoring of visitor effect in their own studies, such that nuanced crowd characteristics can be more readily observed, which may provide a more robust image of animal well-being. Thank you for listening, and I'll take your questions.
Good afternoon, everybody. I'm Claire Toinon, and I will talk about how maternal deprivation affects goat kids' stress coping behavior. It is a routine practice in the dairy goat industry to separate a young from its dam soon after birth and to raise the kids with same-age peers. As there is very limited literature on the effect of such practices on goat kids' development and welfare, we conducted a study to see if maternal deprivation influenced or not the kids' stress coping behavior. To do so, we raised 20 kids artificially. We separated them from their dam at 3 days of age and raised them together in a pen so that they were visually isolated from any other individual. We fed them fresh goat milk 3 times per day, and we gave them enough quantities so they would reach complete satiation after each meal. We compared these artificially reared kids to 20 dam-reared kids. These kids were raised in the lactating herd with their dams, but also with other kids and other adult goats. These dam-reared kids fed via ad libitum suckling. When they were about 1 month of age, the kids were subjected to a series of behavior tests. Each kid was first subjected to a novel arena test for 3 minutes. Then we brought a nulliparous goat next to the testing arena for 3 minutes for what we called the novel goat test. Afterwards, we brought the nulliparous goat back to her own pen and we placed a novel object in the middle of the arena for 3 minutes. The tests were video recorded to observe the kids' behavior such as locomotion, activity, and how they reacted toward the novel stimuli. For the results, artificially reared kids bleated and reared on the walls more than dam-reared kids. They also spent more time in contact with the novel stimuli than dam-reared kids, but artificially reared kids groomed and stood idle less than dam-reared kids. So for these 5 behaviors, tests had an effect on the kids' reactions, but the difference between treatments was consistent across tests. Artificially reared kids were also running and jumping more than dam-reared kids, and artificially reared kids also urinated less, took less time to make physical contact with the novel stimuli, and gazed less at the novel stimuli than dam-reared kids. Altogether, artificially reared kids' behavioral arousal was higher than dam-reared kids. They reacted more actively than dam-reared kids, who reacted more passively. The maternal deprivation and the rearing system here had an impact on stress coping strategy. If you want more detail about this study, it has been published in Physiology and Behavior under the same title as this presentation. Thank you for your attention.
Great. If all our speakers can put your video cameras and microphones on, please. Thank you all for your presentations. Okay, so I'm going to try and ask the questions in the order of your presentations. So first up for Sophie. Elizabeth Carter asks how the level of pup cannibalisation in the laboratory compares to the cannibalisation in the wild.
Yeah, it's difficult to tell because, sorry, yeah, do you hear me? Okay, it's difficult to tell because we don't have numbers from the field, from wild mice. So there is studies about cannibalism, but they don't provide the numbers, and it most of the time it's under experimental condition anyway. So I cannot tell you more.
Thank you. And Hugh College asks, some research suggests that neonatal mice may be so developmentally immature that they lack the capacity to suffer in the first few days of life. Does this mean the cannibalisation is not a welfare issue?
Yeah, cannibalism is not a welfare issue because pups are already dead for other reason. I didn't have time to specify it in the presentation. We differ cannibalism from infanticide. Infanticide is the act of killing, and if it was too frequent, it would be a welfare issue in breeding. But infanticide is not so frequent, so it's not a problem. But the problem is cannibalism. So cannibalism is the act of eating a dead pup, and that is not a welfare issue per se. The welfare issue will be why the pups die, and we don't have answer. Or, I mean, we have cues, but we cannot. We still have to investigate it. But the act of eating a dead pup is not a welfare issue, but it's a problem for mortality estimation in breeding facilities.
Thank you very much, Sophie. For Katie, Paul Rose asks, says, nice talk. Are favoured resources always present in the same areas of the primate enclosure so that they will be used by the primates irrespective of visitor presence? Or was there some variation in the exhibit layout to determine any overall potential visitor influence?
Yeah, thank you for that question, Paul. In this study that I was sharing, it's actually over a year of data that we were looking at. And so there's variability in where resources are for sure, both in the visitor present and visitor absent conditions. I don't have any reason to think that there's systematic differences between visitor present and visitor absent conditions. And that's something that Alex Evans, she's the master's student of mine who's looking into this very closely, that we'll look into before we finalize anything. But yeah, I don't think that would explain how we would get sort of this similarity between visitor present and visitor absent findings.
Okay, thanks. Berta Nielsen asks, in the example of provisionist trade, you're making the environment more, therefore reducing the choice available to the animals. Should that be taken into consideration?
Yeah, fantastic question. So we often provide as much choice as possible. I think when we see, as we did in the case of the Takins, that the choices are being provided but not utilized, then, and in that case, I mean specifically, we have less sun or less shade than the Takins preferred to use, it appeared. So we wanted to respond to that and provide them with the resource that they appeared to prefer but not have enough of. And so we responded in that way, but we always try with sort of our program at the zoo to be continually monitoring behavior and space use. We're lucky to have a team of people to help do that. And so we can sort of respond accordingly as preferences change. But yeah, the more choice, the better, but they have to be choices that the animals, want to use.
Thank you. For Vicky, Stephen Wickens asks, does your study simply tell us that keeping rats in a more complex environment is better for their welfare and that laboratories are unlikely to be able to meet these needs?
I think that's a very difficult question to answer without having an absolute measure of welfare. I mean, we did find that rats were performing lots of behaviours that they didn't perform in the lab. So lab conditions are definitely potentially, well, they're potentially much more restrictive, in which case you would expect lab rats to have poor welfare, but without doing a nice study to directly compare their welfare, it's difficult to say.
Thanks. And Jamie asks, is curious about the effect of predator exposure on pinning and other rat behaviours, and whether there is any indication that pet rats can habituate to living with a predator. Have you looked at where short-term and long-term cohabitation have the same effects?
So we didn't have that data, but people have looked at habituation to predator odours in laboratory rats, and the evidence is really conflicting. So there are some papers that will say rats don't habituate at all, and other papers that say rats habituate very quickly. So the jury's still out on that.
Thank you. So Sarah, you were looking at the difference between January, February and March, April. Is there any difference in terms of the length of time the monkeys spend outside in those months, or is there, and would that affect their performance?
Yeah, Chicago, obviously, or maybe not obviously, gets pretty cold in the winter. So, you know, anecdotally, keepers can decide whether or not to keep them in. So if there's a series of, you know, several super cold days, of course, they're going to be inside and they can't work. So anecdotally, yes, there could be a difference. But during our study period, we didn't, as far as I recall, we didn't have any of those days, I don't believe. So they basically had access to the yard whenever they wanted.
Thank you. So I'm just scrolling through questions. I've got a couple of questions for Claire. Stephen Wickens asked, what do you think it means that the dam rig kids are more passive in their coping strategy?
Yeah, so that was the basic conclusion of the experiment. Yeah, the thing is, so we had only one time point, so we can't say anything about the coping style, but for the coping strategy, that was our conclusion.
And a question from Benjamin Lecourt. It seems that your results suggest that individually housed kids are less fearful, but would you say that they were more interested by novel stimuli, maybe because they were deprived of experiences due to housing?
Actually, there was no individual housing, so both treatments were grouped housed. It's just the presence or not of adults. So in DAMRED kids, the kids had access to a more diverse social environment. So they had in their home pen, so they were the 20 focal kids, but also like something like 60 adult goats and other kids that were not part of the experiment. And their enclosure was a bit bigger as well, but otherwise they had access to the same environmental enrichment as the Archidria kids. So yeah, that was basically known except the drinker and the deep litter straw.
Okay. Thank you for clarifying that.
You're welcome.
Yeah, that's all the questions I have. So thank you very much for all your participation. I'm going to hand over to Hugh Gollage for the final part of this session.
Thank you, everyone. Really fascinating set of talks again. I just wanted to quickly wrap up the meeting for today and say thank you to everyone who's presented their fascinating work. Congratulations again to the Medal and Early Career Researchers of the Year. And thank you very much to Claire and the other session chairs for handling the questions so expertly. Just one other thing to note, if you've tried to use our book discount code and it hasn't worked for you, you could try the code VET20, VET20 instead. Apparently there may have been a problem with the one that we gave you. We'll get that fixed as soon as possible, but VET20 should work for you and give you your 20% discount. If you'd like an even bigger discount, of course, if you join UFAW, you can have a 35% discount. So maybe head over to the website and join if you want to buy some of our books at a cheap price. And could I suggest, if you're still not exhausted of animal welfare science for the day, that you go and look at the poster session. There's 119 posters to check out over there. It'd be really great if you check those out and vote for your favorites and leave some comments. And with that, I'll say that we'll see you again tomorrow morning. And don't forget that tomorrow starts at 8:20 UK time. I know that sounds early, but the plan behind this was that we'd have the 2 days running over slightly different times to make it slightly more amenable for those of you who are in some of the different time zones. So hopefully tomorrow morning works a bit better for our Australian colleagues and so on. So we'll see you hopefully at 8:20 tomorrow morning, and thank you very much for attending today. I hope you enjoyed it as much as I did. Thanks very much.